हेलो
जी सर।
जी हेलो अस्सलाम वालेकुम I welcome all the participants in our today's seminar, and this seminar series is a regular part of our dash event calendar, and it's being held on every Thursday at 2 p.m. So today the topic of our seminar is mitigation of melanogenesis and escalation in rejuvenation by Korean red ginseng extract. And our today's presenter is Dr. Evelyn Sabha. She is assistant professor in the Department of Veterinary and Biomedical Science. Over to Dr. Evelyn Sabha, please. Hello. 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 Madam, can you please hear me?
Yes sir.
Please go ahead, please.
Okay, thank you so much, Dr. Farooq. My name is Dr. Evelyn Sabha, and I am assistant professor at the Faculty of Veterinary Sciences, and my specialization is veterinary physiology. I thank you, the Directorate of Advanced Studies, for providing this opportunity to provide research works. The topic I'm going to present today, that is mitigation of melanogenesis and escalation in rejuvenation by Korean red ginseng extract. So basically, before I move further, this research was one of the topic during my PhD. Okay, so let's first talk about ginseng, because the topic of my research has been ginseng. So I just want to familiarize all of you about ginseng. Basically, Korean ginseng is called as Panax ginseng CMAR. So Panax is basically a Greek word, and it means cure for all. Panax ginseng, it is a wonder herb that has been used in Eastern Asia over thousands of years, and botanically it belongs to Araliaceae family, and there are numerous ginsenosides and gintonine fractions which are present in this plant. And Panax is one of the approximately 60% of plant genera, the classical disjunct East Asian and East North American distribution. So the true ginseng is mostly cultivated in China, Korea, Russia, and Japan. We can say the important feature of this ginseng is it is a slow growing perennial herb. I mean, if we just grow and if we want results in a really faster pace, we really cannot, because we have to wait for 5 or 6 years until this plant gets matured, and then we can use its roots for its various beneficial effects. Okay, so according to the regions, there are different names for ginseng. Korean ginseng is called as Panax ginseng. The one which is found in China is called as Panax noto ginseng, and the one which is found in America is called as Panax quinquefolius. So while I am talking that there are many good and beneficial effects of ginseng, and these effects are manifested as in the reduction of fatigue, in multiple neurodegenerative disorders like multiple sclerosis. It is also good for memory and cognition, and it is also very much, you can say, pronounced for the diseases which are related with reproductive system. Other than that, there are also many good effects of ginseng, like in case of the diabetes, in case of hypertension, hyperlipidemia, inflammation, and so on. Okay, so because the research that is related with the melanogenesis or the formation of melanin, or what happens when there is extra exposure. So melanin, as we all know, that is a pigment which gives color to the skin, and it is being produced by the cells which are called as melanocytes. The process of melanin formation is called as melanogenesis. So because my next study is related with the molecular work, so therefore I want to tell you a little bit about the physiological mechanism by which this melanin is being produced. So alpha MSH, which is the alpha melanocyte stimulating hormone receptor, which is MCR1, and that is going to activate it is going to convert adenosine triphosphate into adenosine monophosphate, and that will activate a series of pathways such as protein kinase B, C, reactive element binding protein. So when these pathways are activated, the nuclear factor, which is the microphthalmia associated factor, it gets activated inside the nucleus, because these are the nuclear factors. Once MITF gets activated, a series of proteins like tyrosinase related protein 1 and tyrosinase related protein 2, these are activated. Now, when these are activated, there is the formation of two types of melanin products, that is eumelanin and melanin. Eumelanin is the one which gives us the brown or the darker colored pigment, and pheomelanin is the one which gives reddish or yellow colored, we can say, the color. So now from this pathway, we know that the rate limiting step for this melanin production are these three, which is in fact these four, then all of the four nuclear factors, which is the MITF, the tyrosinase, and the other two related tyrosinase related protein. So any drug, either that is of allopathic in nature, that is herbal in nature, or having any other origin, we have to check or we have to elucidate that at what point it is affecting these components, and furthermore it will reduce or enhance the melanin production. Okay, the next is skin aging, because we are checking the effects of ginseng in the melanogenesis, that is the formation of melanin and the skin aging. So firstly, we have to know that what is skin aging. That is classified into two types. The first type is intrinsic or endogenous aging. That is inevitable, which means that as we get old, our skin, it starts aging, and the symptoms and the features are fine lines, dry skin, and the elasticity is removed, skin sagging occurs. The other type is photoaging, or that is also called as exogenous aging. So photoaging is the phenomena that all of us, including the humans and the animals, we are constantly being exposed due to abundant sunlight, and because of that there is abundant ultraviolet radiation. So you might, most of you might have already heard that, while you go out in the sun, you should apply some sort of a sunblock that is available in the form of creams, lotions, sprays, etc. So why we need to take this? Because the ultraviolet radiations, they are harmful for the body. They are not good for the body. So we have to save the skin, our skin, from the harmful ultraviolet radiation, because they cause very rough and dry skin, they reduce the elasticity, and they are responsible for the formation of deep skin wrinkles. Okay, so keeping in mind all of these things, and also to the fact that the skin creams that are mostly being manufactured and advertised, they are having some sort of chemicals in them. So the scientific community, since a long time ago, they are shifting towards the products that are more herbal based, and they have a very less or very decreased chances of untoward effects. So therefore we sought to find the effects of that Korean red ginseng extract and its cream on these parameters which are related to the melanogenesis and which are related to the photoaging. Okay, so the methodology that we used for this research comprised of three parts. The first part was the in vitro study, and the in vitro study means the study which is done on the cells and not inside the animal or a human body. So it comprised of two tests: the cell-free tyrosinase inhibition assay, which was done in which we saw the effects of Korean red ginseng extract on the tyrosinase enzyme function, because as I told before that tyrosinase is the rate limiting step for the melanogenesis. The other test was melanin inhibition assay. This test we did on the B16F10 melanoma cell line, that is a murine melanoma cell line, which is routinely used to for the studies of melanogenesis and hyperpigmentation and photoaging. So we checked the levels of melanin after the treatment of cells with the Korean red ginseng extracts using a spectrophotometer. After we got the prelim results in the in vitro study, we moved towards in vivo study. So in vivo studies, we used HRM2 mice. So HRM2 mice, it's a modified hairless form of mice, which is also extensively used for the studies which are related to this melanogenesis and hyperpigmentation. So in vivo studies, first of all, we exposed the mice to artificially induced ultraviolet B irradiated hyperpigmentation and phototoxicity for 5 weeks. And during those 5 weeks, we also supplemented the mice with the KRG extracts and with the commercially available sunblock as a positive control. And we checked these different parameters like skin wrinkle measurement, that how much wrinkling was being produced as a result of ultraviolet, and how much it has been treated with the commercially available sunblock and with our test extract, which is the Korean red ginseng extract. We also checked the levels of melanin pigment inside the skin of the mice, and then we checked the expression of MMP9 protein, that is matrix metalloproteinase. Now, in the next slide, I will tell you what is the reason that why should we check the matrix metalloproteinase expression. Later on, for we also did histological parameters for skin, which was HND, that is hematoxylin, and eosine staining for the thickness or epidermal thickness of the skin, and Mason trichrome staining for the presence of collagen that is present inside the skin. We did the real-time PCR for expression of various genes which are related with the this photoaging pathway, and also real time was done for the transcriptional expression, and Western blot analysis was done for the translational expression of those genes. Our third step were the human trials, because at the end of the study, I'll show you that how this study was being incorporated on a commercial basis. So once we got the fruitful results from our in vitro and in vivo study, we moved towards the human trials. And for that, we firstly prepared a cream which was 3% red ginseng cream. We prepared that cream in sweet almond or sunflower oil, and we added tocopherol in it. And the procedure was it was very simple procedure. And once we prepared the cream, we tested for its pH, we tested for its color, and we tested for its reaction. For example, if we put anything on the skin, firstly, we have to think that either it will cause some reaction, for example, some inflammatory reaction or some allergic reaction. And after the cream had passed all these tests, we then tested it on female volunteers. So we measured the level of oil content in them using a sebometer. We measured the water content using corneometer. We measured the elasticity using cutometer, and we measured the melanin content using maxameter. So all these meters like sebometer, corneometer, cutometer, maxameter, These are digital devices that are extensively used these days in cosmetology. Okay, so first I'm going to show you what we found out in our in vitro studies, that is in a cell-free system and in B16F10 cells. So Korean red ginseng extract, we abbreviate it as KRG. It inhibited the tyrosinase activity, which is the mushroom tyrosinase. Okay, this is the picture, which is the graph A. This graph A is showing you the inhibition of the tyrosinase activity, because it is the enzyme tyrosinase that converts L-DOPA into the melanin. So if the tyrosinase is suppressed, there is not going to be the conversion of L-DOPA into melanin. And according to our results, the in vitro results, the highest dose of KRG, which was 1 mg per ml, it significantly reduced the amount of that tyrosinase when compared with when we did not with the control. And this Ka here, it means kojic acid, because in any study, either that is in vitro or that is in vivo, we have to compare it with some sort of a positive control, any drug or any extract which is known for its anti-melanogenic or anti-inflammatory or any other activity. So that was kojic acid. Okay, this first graph was in case of a cell-free system. And for the melanin content, we plated the cells which were the B16F10 cells. And then we treated them with, firstly, we stimulated them with alpha MSH so that they should start producing the melanin. And then we treated them with the 2 doses of K1, means Korean red ginseng extract, which is first dose, and K2 is the higher dose. And also the kojic acid, which is the positive control. So we treated the cells, and after the prescribed protocol, we took the cells and we measured the melanin content in them, and we compared it with the standard melanin curve using a spectrophotometer. So according to these 2 results, there was a suppression in the melanogenesis by Korean red ginseng extract. So our next target was to see that, okay, there is a suppression in melanogenesis. How? So we have to see all those transcriptional factors that are involved with that pathway. As I told in the introduction, that MITF, TYR, ERP1 and 2, these are the factors which are downstream of the alpha MSH signaling, and that is how melanin is produced. So we check the expression of these downstream factors transcriptionally using the PCR technique and translationally using the Western blot technique. These are the gel images and these are the blot images, and these bar graphs are the quantification results of these images that we obtained. So according to this result that we obtained, we saw that there was a dose dependent decrease in the melanogenesis, and all the factors which were downstream of that alpha MSH signaling pathway, all of them were being inhibited, keeping the kojic acid as the positive control. So this the cell-free system, then the cellular system, and then the transcriptional and translational system, it showed us in vitro a rough picture of the anti-melanogenic effects of the KRG. Okay, so because usually during the research, all of the steps that I told, we include them in the in vitro part, and then we go for the in vivo study. So in vivo studies, we used the HRM, which is the hairless melanin possessing mice, which are specific models for the melanogenic studies. So in this study, we gave Korean red ginseng to the mice, and then we measured their body weight and their food intake. So why did we measure the body weight and food intake? Is because we exposed the mice to the ultraviolet B radiation, which is abbreviated over here. SB means sunblock, which is a commercially available sunblock, and these are the 2 doses of the Korean red ginseng extract with the ultraviolet B exposure to the mice, because the exposure to ultraviolet B is itself a stress. Therefore, it is very important to determine that if there is any effect on the food intake of the mice during this process. So while we see that there was no significant change in the body weight of the mice, however, the food intake, when we compared it to the control, it was increased for the commercially available sunblock and for the higher dose of the KRG, which means that KRG, not only it would affect the melanogenic pathway, but also it would help the mice to not lose their appetite. Okay, next we check the expression of wrinkle related genes in the ultraviolet B irradiated hairless mouse model. So I told you previously that MMP2 means matrix metalloproteinase 2. Okay, this is a type of protein which is present in the matrix of the body of the tissue, and this is directly related with the collagen. So if the matrix metalloproteinase level is increased, that is in case of the ultraviolet B, it causes the destruction of collagen, and which is what we saw over here, that in the group which was given just the ultraviolet B radiation, the MMP level was found to be very much high, and we determined it with the use of ELISA from the proteins extracted from the skin. Okay, and with the treatment of KRG and our positive control, which was the sunblock, it was significantly reduced. And the similar expression we saw at the transcriptional level for MMP2, MMP9, and interleukin 1 beta. These three components are directly related with the UVB induced collagen destruction and damage. Okay, next we check the effects of KRG on the melanogenesis in the UVB irradiated skin, because like I told you, when we give the ultraviolet radiation, or when we ourselves are also exposed to it, our skin starts tanning. The color becomes more darker. The color becomes more darker because more and more this melanin it starts producing. So we check the level of melanin, or we can say we check the level of melanogenesis because of this ultraviolet B treatment. So we treated the mice with the toxic level of ultraviolet B radiation, and you can see here the graphs, there are two sub bars. So one bar means the right side of the mice, which is this one, and the other side means the left side. So the right side of the mice is the one which we treated with the either with the sunblock, in case this is the sunblock, and this is the lowest and this is the highest dose of KRG. So at first week, okay, we treated the mice for 5 weeks with ultraviolet and with the sunblock and the Korean red ginseng. So in the first week, we did not see any difference in the melanogenesis or the pigmentation. As you can see, it looks very much similar as compared to the ultraviolet B. But in case when the third week came, we started to analyze, we started to see some little bit more pigmentation in case of the ultraviolet B, which is the control group, and it was a little bit less in the sunblock and in the highest dose of the Korean red ginseng extract treated group, which is shown here that there was a little significance as compared to the ultraviolet B group. And then in the fifth group, the highest level of significance was observed. And you can also see that the color looks like a bit more refined as compared to the first and the second week. So thereafter, we can say that Korean red ginseng extract, as compared to when we do not put it, as compared to the UVB ultraviolet B control, has reduced melanogenesis, which means it has reduced the level of pigmentation that is induced by the harmful ultraviolet B rays. Okay, so previously I told that there is a wrinkle formation, in fact a deep wrinkle formation due to toxic ultraviolet B radiation. So we checked the formation of wrinkles in the mice. Also this experiment we did on 3rd, 4th and 5th week. As you can see here, in the UVB, the mice which were given the doses of ultraviolet B radiation, there is extensive wrinkling on the skin of the mice, while with the treatment of this commercially available sunblock, of course, and with the highest dose, the amount of wrinkling was very less when we compare to the only UVB exposed mice. Okay, here is the mean depth of the skin wrinkles. So this test we actually did. We took the pictures and we did the image analysis. Also, we used one of the that is called as patches. The name is some double-sided patches. We put it on the skin after anesthetizing the mice, and then after some time we just pull the patch and we see the amount of wrinkles on that. And according to that, we made a bar graph, and you can see here that at the these 3 bars show 3 weeks, and at 4 weeks, and at 5 weeks. And then with the treatment of the commercially available sunblock and with the KRG, which is our test extract, there was a significant reduction in the wrinkling of the skin in the mice as a result of ultraviolet B radiation. Okay, so as I told before that the UVB not only increases the pigmentation, wrinkles, but it also increases the thickness of the skin, which is not a healthy sign. And the collagen level is deteriorated because, as previously shown, because of the matrix metalloproteinases. So we checked the epidermal thickness and collagen levels as well in the skin of the experimental mice. And we did H hematoxylin and eosine staining for finding out the epidermal thickness. And what we found out that these are the pictures, and then these values, for example, this is 22.13. So this 22.13 has been shown in the bar graph over here. 22.13 micrometer was the thickness of the group which received harmful ultraviolet B radiation, and this 8.5 was the thickness of the mice which were given the KRG at the highest dose. So there is a significant difference even numerically and also in the graphical shape, which means that Korean red ginseng decreased the epidermal thickness that usually occurs as a result of harmful ultraviolet B exposure. Secondly, we check the level of collagen because this is a very common, everybody might have heard a lot of time that, and there are many cosmetic products both for males and females which say that there is a high collagen level in it and it will rejuvenate your skin, it will repair the skin. So collagen level is very important for a healthy skin, and that can be found out using the MT Mason trichrome stain. It stains the collagen fibers blue. So you can see here that in the UVB, in the harmful UVB treated mice, not treated but exposed mice, the intensity of the staining is very less. If you compare it with the group which was given the KRG at its highest dose, which means that the level of collagen degradation was reduced by Korean red ginseng extract. And thus, we can say that it somehow works with the rejuvenation. Okay, so I have made a summary of the experiments for the in vitro and in vivo. The Korean red ginseng extract, we checked in vitro on the B16F10 cells, and we checked on the, this is what, how the HRM2, the mice, they look like this. Okay, so we saw in the previous overheads that there was a decreased or downregulated expression of all of these transcriptional factors. Moreover, in the in vivo studies, there was also downregulation of all these factors, which is the MMP2, 9, interleukin 1 beta. The wrinkle formation was very less, epidermal thickness was reduced, and collagen degradation was very much decreased. Okay, this is the study that we got from our you can say cellular and animal experiment. Now we wanted to check if we could get a permission for human trials, and luckily we did. So we moved on towards the human trials. And in case of the human trials, we used 10 healthy female volunteers, and we divided into two groups. So two groups mean 5 groups were control, which used the cream just as their normal cream, which was not having ginseng. The other 5 were the experimental group, which used the 3% ginseng cream as a part of their daily beauty routine. So I told you before that firstly, even if we make any sort of a formulation, we have to check it for its sensitivity, for its reactivity. So for the discoloration, we did not find any discoloration in the cream from day 1 to 30, as you can see over here. Moreover, the pH is slightly acidic, and it was almost the same during all that 1 month of the experiment. Moreover, we checked, we did the patch test, and these are the scorecards for that patch test, which is the irritation, for the edema, and for the erythema. So when we got these scores, then we quantified them later on. But anyways, we did not get any of the side effect or any of the reactivity due to this cream. So next, we gave this cream to our volunteers, and they used it for 30 days. And how did they use it? They cleansed their face for 1 hour, and then they put this cream at night. So we checked the level of resilience. So resilience means resistance, means elasticity, how much the skin is elastic. Either the cream or anything which is present in it has got some properties that can increase the elasticity or not. That is called as resilience. So this is the picture. This is basically a vector picture. And we told them to put the cream on the T zone, which is this one, and then on the U zone, which is number 2. Okay, so resilience was checked at different, which is the forehead, at the right cheek, and at the left cheek. And finally, when we see the average, the percentage of the resilience was high in the experimental group, means the group which it was using the 3% red ginseng cream, as compared to the control group, which just use the cream which does not have a ginseng in it, which means that ginseng indeed increases the skin elasticity. Okay, next we check the oil content. Now oil is important. It is we can say that it is a type of a protection barrier as well for our skin against different type of pathogen, and our skin normally produces oil in the form of sebum from the sebaceous gland. So what we found with this experiment was that there was 67.8% increase of the oil content in the experimental group, which received the ginseng treatment, which is more high than that of the control, which was 45.6%. Okay, next we check the moisture content in the skin, because moisture and oil, they both balance each other, and that's how the skin looks more, the skin glows and it looks more appealing. So we checked the moisture content in the skin, and it was found at a negative of -14.5, which is a more negative value as compared to the control group. So why we have got this one? Because the experimental period for the human trials in this research was October and November, and October and November are mostly dry winters in the South Korean region. So that is why there was more moisture loss in this parameter. Next, we check the skin tone in the experimental group and in the control group, and that is perhaps the most important reason of the cream that we are using. That is, the skin tone should be it should be less and it should be normal. So we can see here clearly that the value of skin tone is -8.24, and that of the control group was 2.12, which means that the Korean red ginseng 3% cream, it reduced the level of melanin, and that is why the skin tone was towards normal or towards a more lighter side as compared to the control group. Henceforth, the skin whitening effects. Okay, we also measured the level of erythema in the skin. So erythema is basically minor patches of inflammation that happens when you are using some cream, and every person's skin is different. So the level of erythema was very much reduced as compared to the control group. When we see the difference, the control group is minus 3.6 and that of the experimental group is minus 33.8, which means that, which also proves one of my another research prospect, that ginseng has got anti-inflammatory effects as well. Okay, so that is all that we were done with the human trials. Of course, there are further more advanced trials are still going on, but conclusions of this study was that application of this KRG based cream, it reduced the wrinkle formation and it also reduced the dermal thickness, and it also it did not have an effects on the degradation of collagen. Moreover, as we all know that collagen is accepted for its skin elastic properties, and when there is extensive exposure to ultraviolet radiation, there is the matrix metalloproteinases, they degrade the collagen, and there is something we have to give from the outside, either in the herbal form or in some any other drug form, that should reduce the degradation of collagen, and that's what the KRG extract and the KRG cream, it had anti-melanogenic, it had skin whitening, it had anti-inflammatory effects to the skin which was exposed to the ultraviolet radiation, and the KRG, it decreased the levels of that harmful MMPs, and it also have good effects on the collagen, and the wrinkle formation was very less, the pigmentation level was also very less. And as we say that when we do the research, we are supposed to, especially this type of research, when we have the human trials, we should go for the commercialization, or that extract or any of the thing like that, it should be present for the common use. So according to this study and many other studies, I have also another study on the red ginseng oil in melanogenesis. So there are many, these are four, but there are also many others who are using this red ginseng in their creams, and these are commonly available for use in the South Korean regions, and these are also available for if you want, you can also have it from some international sellers. So according to this research, the product name was this one. So that's it from my side. Thank you so much for your attentions, and if any one of you have some comments, some question, and also some future collaborations regarding this, or regarding anti-inflammation, or regarding any biological activity, they are highly appreciated and encouraged. Thank you.
The house is now open for question and answer session. If anybody has questions, you can ask the question. If there are no questions, then I would like to thank Dr. Evelyn Sabha for delivering a very informative lecture on the subject. And I would also like to thank all the participants for their participation. Thank you.